It started to find that people have started.
I know. That's why I say close to I just don't have contact with the book. I'm sending it to my son-in-law. The other one is copy piano.
Ja, senti molto piano
Also to review.
Hey, good to see you.
Talk about Austin newscast. Come on, babe.
Good to see you. Really good. Yeah, why not?
No nasty questions, Ben.
What kind of reputation do I have?
I told him to take the stairs, you know.
Otherwise, you're standing in the middle there.
It's pretty straightforward out here. Yeah, but I just don't know where.
You want that much?
No.
We have a bunch of.
Especially if. Bodies.
I was walking over.
Oh, it's Simplex.
Pretty aggressive, like one.
We have a lot of work to do.
The way I like my machine. Covered everything that needed to be covered.
Yeah, it is. Yeah, it is.
You took the fastest. Not today. Oh, well.
I think we'll get started. So it's a great pleasure to introduce Matt State. And you probably all know him. Matt received his MD from Stanford. Then he did his residence and the fellows at UCLA. Then he did the writing. With no disrespect to MDs, he decided to get his PhD, do the writing. So he actually went to Yale, did his PhD in genetics. I never heard of things like that. He realized that if you really want to understand doing research the right way, you need to get a PhD training. So for all graduates, you are doing the right thing. And these, and so PhD, but also carried over for all throughout his career. He actually he is one of the most prominent leaders in psychiatric genetics and around the world. And all the study he does, he want to do the right thing and want to use the most robust, the most creative ways and to solve the most difficult problems. So these have been showed by many of his research discoveries and also by his leadership position in many of the international large-scale genetic efforts, including NIH-sponsored autism sequence consortiums and many others also heavily involved in the Simons Foundation. So these have been heavily, really heavily paid off in the last few years, and we're very glad to have him here. I want to say one last thing. Not only Matt is a great scientist, he's also a great person. My old advisor always said, it's very easy to find a lot on the street, very easy to find a lot of great scientists. Also very easy to find a lot of great people, but it's really hard to find a great scientist and a great person. So, Matt.
Yes.
Back at you, Guoping. Did someone clap when they heard I got a PhD? I thought. All right, so I'm thrilled to be here, and looking around the audience, it's hard not to think of bringing coals to Newcastle, but I appreciate you coming, and I'm going to tell you today a bit about what we've been doing in the lab, some recent progress in autism genetics, and really I wanted to spend some time talking with you about how we've been thinking about sort of what the next steps are now that we've had some success in gene discovery in autism, what some of the conceptual challenges are and how we're trying to approach that in the lab. So quickly, some disclosures. I don't make any money from any of this, so there's really nothing that is terribly pertinent. Autism spectrum disorders. So autism, neurodevelopmental disorder, appears in the first months of life, is generally fairly stable across life, and is characterized by fundamental impairments in social communication, as well as repetitive and restrictive behaviors. That's the new diagnostic formulation in the DSM. The core deficit of social impairments has lasted throughout, from the earliest descriptions of autism. And while many of you may think that this is a really vague way to think about diagnosing a human disorder, and it is, we suffer this across psychiatry where we make diagnoses simply by sort of observing and listing behavior and then coming up with syndromic diagnosis. In fact, I think it's important to know that if you take two psychiatrists and you put them in the room with someone who has, so did anyone hear? Better, okay. Put two psychiatrists in a room with someone who has classical autism, they may not be able to agree on anything, but they will both diagnose the child with autism. So the inter-rater reliability is very high. And what that suggests is that whatever else is true, we're able to recognize a set of fundamental symptoms that we can all agree is sort of profound social disability. We've known for many, many years that genes play a major role. As a general proposition, almost all of the studies, and now there are about a dozen, of the genetics of autism show very high concordance with monozygotic twins, and the dizygotic concordance rate has creeped up over the years, particularly as the diagnosis of autism has broadened to include really all kinds of levels of severity of social communication problems. But the difference between monozygotic and dizygotic concordance rates, so twin studies and family studies all show that the lion's share of the risk for autism is carried in genetics. One of the reasons I went back to do a PhD was because of this third bullet. I was working on units when I was training as a child psychiatrist and see kids who are profoundly disabled. We know very little At that time, we knew almost nothing about the underlying pathophysiological mechanisms, and it's very frustrating in the absence of that we really don't have any effective somatic treatments for the core deficits that we see in autism. We can help a bit with behavior. We can use behavioral approaches to make life somewhat better, but we really have nothing that gets at the core limitations, and particularly in the social domain. So the idea that really started my lab and continues to drive the lab is that a lack of insight into molecular, cellular, and circuit level mechanisms has profoundly limited our ability to think about how we develop new and more effective treatments. And then really a minimalist notion that genes are one way in to solve that problem. So it will get you to the molecular level. So my lab, as a genetics lab, we have not been terribly interested in things like population risk or how to explain all of autism. We've really been focused on identifying genes of large effect that we can then use as to pull on the thread to see if we can understand biology. So this was sort of, I've been showing some version of this slide for now 20 years. And this was the initial idea that we would find particularly a rare mutation. Rare mutations in humans have a tendency to have large effects or larger effects. So we would find a rare mutation reliably, that we would know that that was related to the human phenotype, and then we would pull on this thread by studying expression and function, by moving to model systems, then ultimately by elaborating pathways, identifying treatment targets, and moving forward. And I'm going to come back about halfway through the talk to tell you why, after 20 years of looking at this diagram, I realized it was profoundly wrong, but I'll get there. Okay, so autism genetics. Very briefly, I give you 2 slides on the history of autism genetics before talking about some of the more recent work from our lab. So one thing that I want to point out is that there's been tremendous progress in human genetics in all areas of complex common human disorders. The progress in the genetics of autism has been somewhat distinctive. So when you talk to most people about what's happened in the last few years in genetics, there's really been this explosion of understanding of the contribution of common alleles that are present in a significant portion of the population, and their contribution to the risk for things like schizophrenia, bipolar disorder, diabetes, and a host of other phenotypes. There are hundreds of phenotypes now where state-of-the-art studies have demonstrated that common variation plays a significant role and are sufficiently powered now to be able to identify specific alleles. These 10 common alleles tend to have relatively small effects, particularly in neurodevelopmental disorders. But with regard to autism, unlike schizophrenia or many other disorders, so far, the search for common alleles has not been productive. There's tremendous evidence that common alleles will play a role in autism, but at present, the sample sizes are just been too small for us to be able yet to leverage. Now, I believe this is going to change, which in the next, last week it changed. Okay, so it changed 2 weeks ago. But as a general proposition, I think it's fair to say, and even I'm sure Ben would agree, that 5,000 was not quite enough, and that, and not surprisingly, as we get to larger and larger samples, we're going to be able to identify that. But in contrast to some other conditions, in fact, the search for rare variation has consistently been a thread throughout the history of autism genetics. It has been extremely productive. So this started really many decades ago with the cloning of the fragile site on the X chromosome that then led to the discovery of the Fragile X mental retardation protein. It's been clear for as long as people have had their kind of eyes open to it that there's an increased rate of autism within kids who have Fragile X. And it also turns out to be the case then in many single gene or monogenic forms of intellectual disability that you see increased rates of autism. Studying those things has turned out, as I'll show you, to be highly productive, not just to characterize those quote unquote rare forms of autism, but actually appear to be at least providing evidence that there are important points for understanding more broadly autism in its more typical form, what we see day in and day out in the clinic. The first success is in what you call idiopathic autism or more typical autism. This is autism where there is not an associated intellectual disability syndrome or identifiable dysmorphology, but again, just the kids we would typically see in clinic. Really began in around 2003. Thomas Bergeron's lab in Paris was studying rare families, looking at regions of the genome where they could see that there were deletions under the light microscope, and then sequencing those regions, and found mutations in neuroligin 4X that then led to the identification of neuroligin 3. These are synaptic adhesion molecules that had mutations. Now I think we have real confidence that they're associated with autism. And then there were a handful of others using often similar kinds of approaches, finding regions of the genome that were grossly disrupted, then identifying the gene that was responsible, including SHANK3, Nerexin-1. Contactin-associated protein 2 is somewhat different, but was found in consanguineous pedigrees. It's a recessive form of autism. So what these pointed to relatively early on, just after the turn of the millennium, were that rare recessive and de novo mutations, actually both neuroligin 4X and Nerexin-1 were initially identified, and actually SHANK3 as well, were identified as new mutations. So the observation and the offspring, not in the parents, led to focusing on those regions and then looking for the gene within them. So it really, actually, almost all of the sort of recent progress can be traced back to the Bergeron lab in 2003 with these very initial observations about these kinds of mutations. And really important, but there was a very long period where really this gene discovery was sporadic at best. So my lab was trying to do the same thing that Thomas's lab was doing. We started in 1997, and after many years of looking and trying to find these kinds of rare families, we were really struggling, and actually contributed a bit to contactin-associated protein 2. But apart from that, we almost a decade of what felt like wandering in the dark. So the shift, I think, in the field, certainly with regard to rare mutations, came at about 2007, 2006, late 2006, in which, because of the advances in genomic technologies, it was possible to screen the entire genome for small submicroscopic deletions and duplications of material called copy number variation. And when you could do that in kids who had quote unquote idiopathic autism, so typical autism, it became clear very quickly that you didn't need to look in that many in order to identify that children with autism, particularly if they were the only affected child in the family, were much more likely to have a new de novo copy number variation, the submicroscopic deletions or duplications, compared to controls. Now that was, the initial observation was really important, several things about it. So one, there was a type of mutation that we could detect and where we didn't need huge numbers in order to be able to see a significant difference. And that suggested that the effect size was going to be large. So the first studies were less than a couple hundred people, and you could still see that there was a difference between cases and controls. But the second really important observation was that these did not appear to be randomly distributed around the genome. It did not appear that there was a chromosomal breakage problem in kids with autism, because when we looked, we began to see almost immediately after that first observation by Jacquemont and then Jonathan Sabat in Mike Wiggler's lab, is a series of papers here in which it showed that there were certain regions of the genome in which these were lining up. And in particular, the first that was identified was on chromosome 16, 16p11.2. And what this suggested was two things. Again, one is that the effect sizes were large, and the second was that you could begin to use observation of seeing the same mutation or a very similar mutation in the same spot in the genome to begin to narrow in on genetic loci and ultimately genes contributing to autism. So most of what my lab has done since that point has been a consequence of a major investment from the Simons Foundation. They created something called the Simons Simplex Collection, driven by those early observations that new de novo mutation was contributing to autism. And they put together what at the time was quite unique, but it was a sample that was designed to enrich for de novo mutation. And you can look here and see, so this is just two parents, blue is unaffected, red is affected. And all of the families had this structure. They had unaffected parents that were evaluated both categorically by saying, do they have autism or not, and also by dimensional measures of social ability, et cetera. So to do the best possibly could to say that these guys were unaffected, and then that we were very fastidious about characterizing a child that was affected, and in about 80% of the Simons families there was also at least one unaffected sibling. And when you think about this, it's a pedigree structure that's designed, or it looks like, that it's been hit by lightning. And when you think about de novo mutation, right, the idea would be that if it looked like there was just this something arising spontaneously in the family, that would enrich for the observation of de novo mutations. So it laid out this really beautiful and simple experiment. And the simple experiment is that whatever tool you had available to look through the genome, you could compare the DNA of the parents to the offspring. Anything that was present in the offspring and not present in the parents was at least possibly a de novo mutation, and then you just have to go and confirm whether that's the case. De novo mutation is sufficiently rare that was not a particularly difficult thing to do. You don't see that many observations, so generally, I'm fond of saying that this is really my style, because all you have to do in most pedigrees is count to 1. It's either 1 or 0. So you compare the parent's DNA, the child's DNA, and then the beautiful thing about the Simon sample, again, was that the way that it was designed, then we could compare the rate of those events within families of the affected to the unaffected child. And this was tremendously important, because one of the big confounds in genetic studies is not appropriately matching between cases and controls. So you have to do a lot of gymnastics in order to make sure that you're not looking at differences that are just a consequence of ancestry and not a consequence of the disorder that you're ascertaining for. But when you're working within a family, whatever the ancestry is of these parents, it's going to be randomly distributed to their offspring. And so you can use that essentially to zero out or normalize the influence of ancestry and asking whether or not there's an increased rate of de novo mutation in cases versus controls. So the first study that we did was published in 2011. We've just followed up with the entire sample. So the first study was 1,000 families. That was about 5 times larger than the study of Sabat or Jacquemont. This is Stephen Sanders, who's now an assistant professor at a place out at UCSF, who you'll see his picture many times. Brilliant young assistant professor who's led this work over now almost 7 years in the lab. So the first study was about 1,000 families. We just published in Neuron a paper that now characterized the entire Simons sample, which is nearly 3,000 families. And what you're looking at is the rate of de novo mutation. These are duplications in blue, deletions in red, in probands versus their siblings. And it's divided into the subsets, and the first sample, then the new samples, and then the combined sample, just to show that we're getting consistent results across the separate parts of the Simon Simplex collection. And if you look all the way over on the right, this is the number of de novo CNVs in cases versus controls, and this is the number of genes within CNVs. So we knew from our first study that if CNVs were genic, if they contain genes, that appeared to be more of a risk event than those CNVs that did not carry genes. And so pretty much all this shows is no matter how you cut it for de novo events, copy number variations, you see an excess in cases versus controls. And this is just showing that signal is not present when you look at transmitted events. So all of the signal that we're able to derive in order to discover genes doesn't mean that there's no risk here. There are just many more transmitted events than there are de novo events. And so the signal to noise ratio with regard to de novo mutation is much more beneficial to searching for novel genes. Now, so we've got 2 things. De novo CNVs are more common in probands and siblings. I'm just going to make a quick point here that everywhere where we look, we find that de novo CNVs, particularly those that contain genes, are more common in girls than boys. And I'm going to come back to that in a moment. Now, I told you that from the first observations, even in a couple hundred people, were followed very quickly by seeing that the CNVs were not randomly distributed. And very early on, we worked with Bernie Devlin and Catherine Rader back in 2009, 2010, to think about how we could come up with a rigorous statistical framework in order to be sure that when we were looking at a de novo event, that it was truly related to autism, because de novo events also happen in controls. They're at a greater rate in cases, but you see them in controls as well. And frankly, all that we did is we were able to leverage the fact that these were de novo events. And what I mean by that is that, a de novo event is happening at most at once a generation, typically, the kind that we're looking at. And so instead of simply counting the number of observations in cases versus controls, once we understood enough about the characteristics of the mutation, we could ask, how likely is it that a de novo mutation will happen in the same spot in the genome more than once? And when you use that, you get an awful lot of statistical power because the target is big and the number of shots that you have at it is small. And so actually a relatively small number, certainly compared to looking at common variation, a small number of recurrent events gives you high confidence that you're looking at an associated copy number variant. And this is from 2015, summarizing all of the copy number variants that have now been identified that reach our threshold for genome-wide significance. And we use two different approaches to do that. The only thing I'll point out here is that we've taken to using a false discovery rate as an alternative to a p-value, particularly as I move on to talk about de novo point mutations. So the idea here is just to give you a sense that there are a number of loci. We can actually predict that there are going to be many more than this, but these clearly, after several years of study, are a handful that are carrying very large risks and that are reliably associated with autism. I'll talk more about that in a minute. Now, many other events are seen in the clinic, and there are lots of ways to think about whether or not they're likely to be pathological. This is a list of those from all of the large-scale studies that allowed us to look at de novo variants that reached our statistical threshold. So a relatively constrained view of the copy number variation landscape in ASD. What I want to do is focus actually on just one region, and this is from our first study in 2011. We replicated the finding that 16p11 was associated with autism, and then we identified a new locus, 7q11.23, and we found that duplications in that region led to classic autism, and looked hard at the phenotype to see whether there's anything unusual about it and couldn't find anything. I'm just going to show you a brief video here of a kid with autism for those of you who are not familiar with the clinical phenomena. There's no sound here, and one of the reasons I use is that you don't really need sound in order to be able to identify a kid with autism. So I think you probably saw at the beginning, flapping, which is repetitive stereotype behavior, is very characteristic of autism. And again, you don't need to know this child has essentially no useful language. That the sense about how the child is relating to the adults in the room, that's his father on the right and examiner on the left. This is, one version of profound impairment in social communication. This, for a 13-year-old boy, the way that he exists in the room, I think for most people it's relatively apparent that this is not sort of typical social interaction. So this is a region 7q11.23. There are 23 genes in that region. What I want to show you is what happens when the identical region of chromosome 7 is deleted. And this does have sound. So all I want you to pay attention to is the social relatedness of every kid in the room.
To ask questions, but not as much as these kids.
My favorite color is blue. I have met Barney the dinosaur. I live in New York City.
Do you have any sons or daughters?
I do.
Wow.
I have 2 daughters and a son.
And kids love to make friends, but not like these kids.
What nationality are you?
I'm Italian. Oh.
How do you, buddy?
That's my favorite line, "Ciao to you, buddy." So this is Williams syndrome, and it's been known for a long time that deletions on chromosome 7q, where we found this duplication that leads to autism, lead to Williams. And Williams has a very reliable behavioral phenotype, and you're looking at it. Very, very social, a tremendous interest in reciprocal social interaction, and a variety of other really interesting things that I won't get into. But the reason that I show that is that, and I'm going to show you other data as well. So when we found these CNVs, they are associated, as are other de novo mutations, with decrements in IQ versus what you would expect from parental IQ, which is typically the way that you would predict where a child should be. Okay? And so we definitely see a shift in IQ down when we find these de novo mutations, which have led some people to question whether or not what we're finding is just general failure of brain function that leads to intellectual disability along with social disability. And one of the reasons I like to show this is that while those things often can go together, both of what you saw, classical autism and kids with Williams syndrome. In fact, when we looked across our kids with 7q, there was no difference in overall IQ, but you can still see that as a consequence of these mutations, that they can manifest in profound differences in social reciprocity. And I think what that suggests is that while both may be affected, I think it's important to keep in mind that these still have the potential, we believe, to identify critical aspects of neural systems underlying social behavior as well as just general cognitive abilities. Now, we thought this was really cool in 2011 and even in 2015 to be able to identify specific regions with high confidence that lead to autism, and would be the start of that process of pulling on the thread, but really not the ideal experiment, because the ideal experiment would be to get to single genes as opposed to groups of 20 or more genes, as many of these CNVs contain. And so this became possible. This is just to remind you of how dramatic the progress has been in the declining cost of sequencing the genome. So now genomes at 2015, depending upon where you buy them, are somewhere in the neighborhood of $2,000. And this opened up the possibility of doing exactly the same experiment that I described with regard to copy number variation. Now we could do it with regard first to the exome, so the coding portion of the genome. And we did exactly the same thing. So, again, count to 1. And what happened is that, actually, Ben's up there somewhere. There you are. So pretty much everyone landed in the same spot at the same time, which is that we all found that if you looked at de novo mutations that were damaging, particularly those that resulted at least putatively in a loss of function, that you saw the same kind of overrepresentation that we saw with regard to copy number variation. And we could use basically the same approach to defining the statistics of this to use the recurrence of de novo mutation in a single gene as a way to increase statistical power. So with relatively small samples and relatively few observations, we could be confident that we were looking at ASD genes. So Mark Daly's lab with Ben in the lead, my lab with Stefan in the lead, and Mike Wigler's lab, also Brian O'Rourke, who's formerly a grad student of mine. I like to take credit for him sometimes, who was in Evan Eichler's lab. So, I have to say that there probably are not many psychiatric geneticists in the audience, but just this slide is remarkable to a psychiatric geneticist, because for so long the field was characterized by everyone contradicting each other. It was like, oh, we got a common allele, and then 2 weeks later it'd be like, no, and then everyone would argue about why a non-replication was really a replication. And so it really is, it explains some of my kind of recently found optimism in the field, that now I'm able to stand up and show that really, regardless of which lab you're in, that we're ending up with essentially identical answers. Just like the list of CNVs that I showed you, now I can show you a list. This is from Stefan's paper that was just published. And what we've done is there were actually 2 major papers just before this from one from the Simon Simplex collection, the other from the Autism Sequencing Consortium. And we took all that data and put it together. And then we took the exome data that had been previously published and used a statistical framework that allowed us to combine evidence from copy number variation and from exome data in order to identify autism genes. And so this, again, I told you that we like to use a false discovery rate, and here the false discovery rate is less than 0.01. For some of these, CHD8, SCN2A, it's much less than 0.01. But we also let people kind of take a look at additional genes that we're identifying that are showing evidence. I wouldn't say that it's definitive evidence, but what this is, it allows people to kind of pick what they want to do. If you want to make an animal model, would strongly suggest that you stay to the left. Here, is it the left or your right? I don't know. But if you're doing, thinking about doing a systems analysis or computational stuff, you might want to be including genes that may not have rock solid evidence, but still have a greater than 90% chance of being related to autism. There are just a couple other things that I wanted to point out, and this gets back to this question about what is it that we're finding. So we find these de novo mutations. We found them in the structure of the DNA, and we find them in the sequence of the DNA when they're damaging. And again, this question has come up, and this is just a different line of evidence that suggests that while there may be overlap with intellectual disability, it is not the same thing as intellectual disability, so that's the top panel. And then the bottom panel I just want to talk about for a moment, because I think we now have the power to begin to really ask the question whether or not the large CNVs are going to resolve down to a single kind of smoking gun, or whether it's more likely that there are multiple genes within those regions that are contributing. So what this is showing is nonverbal IQ, and these are the percentage of probands with a de novo loss of function mutation or CNV. And let's see, de novo loss of function, de novo CNV, so what, nonverbal IQ, trying to figure out what we did here. Sorry.
You're tracking the relationship of risk versus IQ.
No, I got that. I'm just looking at what this is. Sorry, percent of proband with a de novo loss of function, de novo CNV. Oh, this just has the cutoff of an FDR point 1. This is any de novo variant, and this is a de novo variant with an FDR, so in that group. And what we show is that regardless of IQ, while you can definitely increase the yield of de novo mutation by looking in kids who have lower IQ, and this reflects what we saw as well just in the early CMV papers, is that you still see an overrepresentation of de novo mutation across the IQ spectrum. And again, this is really just because I think it's when we're thinking about whether or not we can leverage these to understand something fundamental about the phenotype that I'm interested in, since a core feature of that is social disability, it would not be a good thing if we were finding genes that were primarily responsible just for intellectual disability, with kind of an epiphenomenon being poor social function. All right, so some additional observations from these studies. So the vast majority of de novo SNVs were from father, from the first papers that I pointed out in 2012 from Mark's lab and our lab and Brian and Evan Eichler's lab. So these increased with paternal age, a definite phenomena. Everyone has replicated this. It is a small effect relative to the putative increase in the rate of autism in the population. So it gets nowhere near explaining an increase overall in population risk, but you definitely see that 80 to 90% of the de novo mutations are point mutations, not CNVs, but point mutations are coming from fathers. We can predict based on knowing the mutation rate and how quickly we're seeing double and triple and quadruple hits that there will be somewhere between 400 and 1,000 genes. Error bars are pretty large, but the scale is more important. It's not going to be 10, unlikely to be 10,000, somewhere in this neighborhood. We can also just do kind of a seat of the pants look at what the function, the overt function are, the genes that we've identified, and really half of the initial genes that we pulled out are, at least within databases, are identified either as chromatin modifiers or being involved in synaptic function. And so, when we do more sophisticated analysis, using gene ontology, et cetera, that holds up. And then, as I said, the Fragile X mental retardation protein, it's to me fascinating that this thing that we found 60 years ago, and it took a while for people to really appreciate that there were increased rates of autism in this population. But it's really one of the strongest signals. If you're a target of Fragile X mental retardation protein, you are much more likely to be an autism risk gene. And there are other proteins where there's convergence, but this is really one of the strongest repeated findings. We can predict that de novo point mutations will account, depending upon who you are, either for 15 to 20% of individuals presenting to clinic and 35% of females, or about 10% of individuals presenting to clinic and 20% of females. The difference really is that our calculation here in Sanders et al. Was only around loss of function mutations, and these guys are predicting what ultimately a signal will be from missense mutations, where a smaller subset of them are also carrying risk for autism. As I said, there's an IQ shift to the left, but the excess of de novo mutation cuts across the IQ spectrum. I've already told you there's a greater mutation burden in girls. And then there's a nice paper in Nature Genetics that shows that 3% of the population risk in autism is attributable to de novo mutation. And many people get stumped by this because we talk about 15 to 20% of individuals, but only 3% of the risk. And I just want to remind you that when we think about risk, if you think about a normal distribution of risk, we think that if this is risk for autism, that most of what's under this curve is a consequence of common variation. But most of that will not have someone reach a threshold for diagnosis. So what we're looking at with the de novo mutation are very likely, and there are multiple lines of evidence suggesting this, are that these are just larger hits, and even though they account only for about 3% of the total population, they account for a disproportionate amount of the tail end of the distribution of people who come into the clinic complaining of autism symptoms. Now, we also know that this is not Mendelian risk. These are not quote unquote single gene versions of autism. And the reason that we have a good idea about that is that if you have a first child who has a de novo mutation, the chances that the second child will have autism, if the first child has autism with de novo mutation, the second child will not be back at the population-based rate. So if the de novo mutation were typically carrying all of the risk, you would expect that a second child without the de novo mutation would have a 3% risk of autism. But in fact, it's significantly increased. It's not as high as families in which a child is diagnosed with autism who doesn't have a de novo mutation, but what it suggests is that these de novo mutations are acting on a common variant background, and that risk is accumulating in families with the de novo mutation, pushing one over the threshold. All right. So the view from here. We have some wonderful opportunities. As I said, the source of my great optimism. We have coding mutations. We have loss of function heterozygous mutations that are falling in regions of the genome that we can readily interpret. And they have very large effects. So we can estimate in general that the group that was all the way on the right that has an FDR of less than 0.01